Hello everyone, this video covers Chapter 7 in Thompson, Metabolism from Food to Life. So let's get started. We are going to start with the stages of the fed-fast cycle. And we're going to start with the fed state, which lasts about 3 hours after a meal, and the hormone is insulin. So we want to start with that now. And as we go through this chapter, we will go through each one of the steps. So right now we're starting with the fed state. So this diagram right here, again, focus your attention right here on this pink line. So this is the absorptive phase right here. So lasts for about 3 hours. So this is exogenous, which means this is glucose that comes from our diet. And this is glucose used in grams per hour. So we use glucose from our diets in the first 3 hours after a meal. So what the body has to do is it has to keep the blood glucose concentration the same no matter how much you eat, how little, how much you exercise, how little, how rested you are, if you're sick, anything like that. It needs to keep the blood glucose concentration the same. So that's sort of like the motivation here. So we're starting with the absorptive phase after we eat. So this is from our textbook. So this shows a student having a meal, and it's a mixed meal of protein, carbohydrate, and triglycerides or triacylglycerols. So what happens is the proteins are digested to amino acids, carbohydrates, starch, to glucose, and the triglycerides to the fatty acids, 3 of them, and that glycerol backbone. And they'll either be used right away, or they'll be converted to some kind of storage form that you can use later. Again, this is exogenous. So this is starch and triglycerides and protein from our diets, and they undergo catabolic or breaking down reactions, and they were broken down into smaller compounds. So starch is broken down into glucose, and that releases energy. Triglycerides are broken down into glycerol and 3 fatty acids, also releases energy. And proteins are broken down into amino acids. So all of these processes are catabolic or breaking down, and they all release energy. So I had you complete the table on your handout in question 1 using this figure as a resource. So I just summarized the answer just now. Okay, so again, degradation or catabolic or breaking down pathways are food molecules are broken down to smaller molecules. Again, disaccharides and polysaccharides to glucose, fats to glycerol and fatty acids, proteins to amino acids. And these reactions release energy, and we can capture some of that energy to make ATP. And ATP is going to be able to drive other reactions that we need. The other thing that happens is that in these catabolic reactions, the molecules lose electrons and protons, and those are going to be donated to coenzymes, and those coenzymes are sort of helper enzymes, and those coenzymes will also, when they donate their electrons and protons, that's going to lead to additional ATP production, as we'll see as we progress through the chapter. So again, we have these catabolic reactions that this is the figure I just showed you. But what happens is this diagram shows how you can start with adenosine monophosphate, adenosine diphosphate, add phosphate to it, and that's how we get our ATP. So this requires energy, but as you can see from these catabolic reactions, energy is released. So we can use some of that energy and capture it and make ATP. So again, if we're going to focus on this part, let's do carbohydrates first. So we're breaking down starch in our diet to glucose. And what happens is, if we go to this figure right here, glucose is the primary fuel source for all tissues, so we will be using that. But if we have extra glucose that we don't use right away, then we'll store some of that glucose as glycogen in the liver, that's the liver right here, and the muscle. And then if you still have more left, because there's only so much glycogen your liver and your muscle can hold, then you'll convert the glucose to fatty acids and glycerol and make triglycerides, and those will be stored in the adipose tissue. And again, you can use those later. So in question 2, I asked you to identify the products of glycolysis. So what's going to happen now for the tissues that are using glucose as an energy source, that glucose is, this is the reactant. It has 6 carbons. If we go down here to the end, we will form 2 3-carbon molecules. These are pyruvate. So we're going to form 2 molecules of pyruvate. And we're also going to produce as a product NADH and H+. We're going to produce 2 of them. And you can also see that we're spending 2 ATPs, but we get back 4 ATPs. So another product is 2 ATPs. So the answer to this question is, what are the products of glycolysis? Well, glucose goes in, 6 carbons, 2 pyruvates come out, we get 2 NADHs and 2 ATPs. So those are the products. So I think I've said this already. Okay, so glucose is a 6-carbon molecule oxidized to 2 molecules of pyruvate, which is 3-carbon molecule. We produce 2 ATPs, and those electrons and protons that the glucose has, they're going to be donated to NAD plus to form NADH and H plus. This is a coenzyme. It's a helper enzyme, and it's made from the NADH stands for nicotinamide adenine dinucleotide, and you make it from niacin, which is vitamin B1. So that's why vitamin B1 is so essential to have in your diet. Glycolysis, this is a pathway that's found in all cells, and in some cells it's the only source of ATP. So again, when you go from glucose to pyruvate, you get 2 ATPs, and you give the some of the electrons and protons that are given up by the glucose are given to these coenzymes, and we get NADH as our product. So if I'm going to continue on the pathway, basically I've described this right here. So we've taken in carbohydrates from our diet. We broke them down in the digestion process that we talked about back in chapter 3, I believe. And that's mainly glucose. Glucose is going to be absorbed. It's going to travel to the portal vein. The liver will take its share and then pass the rest of this on to the tissues. The tissues will take it up, and glucose to pyruvate, that's glycolysis. It just shows one arrow, but it's actually a 10-step pathway. So what you should notice here is that you only get 2 ATPs, and this is a process called, the next process is anaerobic fermentation. We don't need oxygen for this. So again, digestion, absorption, the glucose enters the cells and then undergoes glycolysis. So what happens next depends on whether you have oxygen available or not. So what happens here is, where I put a box around it, glucose goes to pyruvate, and then pyruvate is converted to lactate or lactic acid. And this is what happens if there's no oxygen and no mitochondria. There's another process we'll talk about in a minute that involves oxygen and mitochondria. But no oxygen, no mitochondria, this is the next step of what pyruvate does. So in anaerobic fermentation, glucose goes to pyruvate. You get 2 ATPs per glucose. That's not a lot. Then the pyruvate gets converted to lactic acid. That occurs in muscles when oxygen is lacking during short periods of intense exercise. But it also occurs in cells lacking mitochondria. And it's actually, if you are a trained athlete, here's another reason to go to the gym, athletic training increases the number of mitochondria. Trained athletes have more mitochondria than sedentary people. So in sedentary people, you get lactic acid even if you're not exercising, even you're at rest. So it's good to exercise to get mitochondria. So this is what happens if you do some kind of short-term exercise. So high jump, power lift, shot put, tennis serve, sprints, things like that. You can get enough ATP from glucose without oxygen. But you can't do this for long. And the other thing is that once you go from, in glycolysis, from glucose to pyruvate and pyruvate to lactic acid or lactate, then that's a dead end. The muscle can't do anything with it. So what happens, I asked you to describe the series of steps here. So this shows pyruvate being converted to lactate. So if we go down here, you go from glucose to pyruvate, that's glycolysis, and ATP is produced, 2 ATPs, and then you go from pyruvate to lactate, and the muscle can't do anything with it. So lactate goes, leaves the muscle and goes into the bloodstream, and then the liver actually can convert it back to pyruvate and into glucose. So what's happening here is glucose to pyruvate, and then fermentation, pyruvate to lactate, and then the liver is able to take to carry out this pathway called gluconeogenesis. The muscle can't do that. So the pyruvate can actually be converted back into glucose through gluconeogenesis, and that can go back to the muscle. You can't do this forever though, because you only get 2 ATPs from glycolysis, but this gluconeogenesis part, that actually costs you 6 ATPs, so you can't do this forever. So again, the muscle can't do anything with lactic acid. The lactic acid leaves the muscle, goes to the bloodstream, and gets to the liver. And then the liver has the enzymes, which the muscle doesn't, to convert lactate back to glucose. And that's called gluconeogenesis. And that requires energy, and it requires oxygen too. So that's what happens for short-term exercise if you don't have oxygen. But what if oxygen is available? Then we're going to do this instead. So pyruvate's not going to be converted to lactate. Instead, it's going to be oxidized all the way to CO2. So you're going to get, so carbon dioxide is as oxidized as you can get. We're going to do a complete oxidation, and that's CO2, which we exhale. So it's going to go through the citric acid cycle. So the citric acid cycle takes place in the mitochondria. So you have to have mitochondria, and you have to have oxygen too. And we'll find out why in a second. So again, if I go through this pathway now, I digested my carbohydrates from my meal. I absorbed them as mainly glucose. Glucose enters the cells, goes through glycolysis to pyruvate. And now, if oxygen is available, you're not going to go to lactate. You're going to go to, you're going to go to acetyl-CoA, then the citric acid cycle, then the electron transport chain, and you're going to get more ATP that way. So I want to show you how that happens. So the citric acid cycle, your authors abbreviate it as the TCA cycle, but it's the citric acid cycle. So if oxygen is available, pyruvate doesn't form lactate. It enters the mitochondria instead and is converted to acetyl-CoA. So in your handout on question 4, I asked you to describe this. So what happens is I have 2 pyruvates, and I undergo a reaction where I produce 2 NADHs, some more of those coenzymes, and 2 CO2s, and now I have 2 molecules of acetyl-CoA. So glucose is, glucose is catabolized into 2 molecules of pyruvate during glycolysis. If oxygen is available, each pyruvate is converted to 1 acetyl-CoA and 1 CO2. But remember, we have 2 pyruvates, so we get 2 acetyl-CoAs, 2 CO2s, and we get 2 NADHs. So if oxygen is available, the pyruvate goes into the mitochondria and is converted to acetyl-CoA. So now we have 2 acetyl-CoA molecules, along with 2 CO2 and 2 NADHs. So this is called aerobic respiration, and this is a summary. So glucose is converted to pyruvate in glycolysis. Then the pyruvate leaves the cytosol, that's the area outside of the mitochondria. It goes into the mitochondria, and then the mitochondria pyruvate's converted to acetyl-CoA, and then it enters the citric acid cycle. And we lose 2 of our carbons as CO2. So what happens next then is now I'm here, and we're going to send an acetyl-CoA through something called the citric acid cycle. And as you take it to the citric acid cycle 1 at a time. So if you have 2 acetyl-CoAs, you have to go through 2 turns. And the artist who designed this actually counted everything up for 2 turns. So in question 6, I asked you to say, what do you get for 2 turns? Well, if you get 2 turns, your products are 2, 4, 6 NADHs and 2 FADH2s, and you also get 2 ATPs. So those are the products: 6 NADHs, 2 FADH2s, and 2 ATPs, and that's what you get when the citric acid cycle makes 2 turns. So for 1 turn, you get 3 NADHs, 1 FADH2, and 1 ATP, but you multiply that by 2 because you have 2 acetyl-CoAs, and it goes through 2 turns. So so I think I said that down here. So protons and electrons are donated to form 3 NADHs, 1 FADH2, and 1 ATP. If you go around the cycle once, you have to multiply everything by 2 because you have 2 acetyl-CoAs, so you have to go through the cycle twice. So to summarize, glucose goes through to pyruvate in glycolysis. You get 2 pyruvates, 2 NADHs, 2 ATPs. If you have oxygen and if you have mitochondria, the pyruvate goes into the mitochondria, so you have 2 pyruvate, and it gets converted to acetyl-CoA. So 2 pyruvates go to 2 acetyl-CoA, 2 CO2s, and 2 NADHs. And then you go through 2 turns because you have 2 acetyl-CoA, so you have to go through 2 turns. And if for 2 turns you get 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s. So what's going to happen now is that we have a lot of NADH and FADH2, and it's going to go in a process called the electron transport chain. So this is kind of complicated, but we don't really need to understand how all the details to understand how this works. So again, glucose goes to pyruvate, pyruvate goes to acetyl-CoA. We go through the citric acid cycle, and the NADH enters something called the electron transport chain. So it passes its electrons and protons all the way down the chain till it finally gets to oxygen to form water, and that's why you need oxygen. So that's the oxygen that we breathe in. And notice that for 1 NADH, you get 1, 2, 3 ATPs. The FADH2 enters a little bit lower, so for 1 FADH2, you get 2. So basically what we're doing is we're capturing some of the energy of this electron transport chain, and we're capturing it to form more ATP. So, and we need to do this because if we're doing aerobic respiration, aerobic exercise, long swim, long race, marathon, long bike ride, hiking, you can get a whole lot more ATP from glucose when you have oxygen available, because you get ATP not just from glycolysis or one turn of the citric acid cycle. And the NADH and the FADH2 that go through the electron transport chain, that gets you a lot of ATP as well. And you're going to need that for long-term exercise. So how many ATP are produced when one glucose is partially oxidized to 2 lactic acids in homolactic fermentation? So that's question 7. It's the same as question 2. The answer is 2. You get 2 ATPs. So now, okay, so let me go back. So that's what happens when you go from glucose to pyruvate, pyruvate to lactic acid, and then you're done. You only get 2 ATPs, and that's without oxygen, and you can do that for short-term exercise. Now for long-term exercise, we're going to completely oxidize the glucose to carbon dioxide, and we need oxygen, and we need mitochondria. So how many ATPs are produced? Well, what you need to do is you have to count up the number of NADH and FADH2 produced. And remember that 1 NADH produces 3 ATPs and 1 FADH2 produces 2. So what we should do to answer that question, which is not on my slideshow, I'll go back here. What you do is you count everything up. So here's 2 ATPs. So if 1 NADH gets 3 ATPs and electron transport, this is going to give you 6. So you've got 2 here and 6 here, so that's 8. And then 2 NADHs again. If 1 NADH gives you 3, then 2 is going to give you 6. So we have 8 here and 6 here. That's 14. And then if we do our cycle right here, here's 2 ATPs. So now we're up to 16. And we're going to take 2 FADH2s, and remember each one gives us 2, so that's 4. So now we're up to 20. And if we have 1 NADH gives me 3 ATPs, then 6 is going to give me 18, and 18 plus 20 is 38. So you get 38 ATPs. That's way more than 2. So you get a whole lot more. You get 19 times more ATPs for the same glucose. So I wanted to remind you that metabolism is the sum of all of the chemical and physical processes by which the body breaks down and builds biomolecules. Catabolism is breaking down, which we have been talking about. Anabolism is building up. So the chemical reactions that require or release energy in the form of ATP, electrons, and protons are catabolic. And we talked about those. So now we want to talk about anabolic reactions. So the catabolic processes released energy, and we captured some of that in ATP in electrons and protons. And now we want to look at anabolic reactions because they require the ATP, but luckily the catabolic reactions gave it to us. So how does that work? So we go back here to our student who is eating her meal, and remember that the end products of digestion, anything she didn't use right away after eating that sandwich would be converted to storage forms, either glycogen or fat. And now is the time to let's figure out how that happens. So the anabolic reaction, so here glucose is built up, to form glycogen. That requires energy. Glycerol is attached to 3 fatty acids. That's also requires energy. Amino acids are used to make protein. That also requires energy. So these are anabolic pathways and they require energy. The small molecules from our food are the starting materials for larger biomolecules that make up our body structure. So we really are what we eat. So the glucose that we from our food is going to be converted to glycogen in muscle and liver. The amino acids from the proteins that we ate will be converted to muscle protein, hormones, buffers, antibodies, transporters, all those protein things in our bodies. And then the fatty acids and glycerol will be used to make fat. And that is stored in our adipose tissue until we need it later. So all of these reactions require energy in the form of ATP, and electrons and protons are required too. But again, luckily, the catabolic reactions got that for us. So in the anabolic reactions that I just showed you, ATP is required. So in this reaction, we show that if we do a hydrolysis of these phosphate groups, that is a chemical reaction that provides energy for these reactions to happen. So we were in the fed state, and the carbohydrates from her food were broken down into glucose, and now we can use them to make storage forms of glucose like glycogen. And again, your liver and your muscle is where you store that glycogen, and your liver and your muscle can only store but so much glycogen, so the rest gets converted to fat. So, again, so anything that we don't use right away after we eat gets converted to glycogen. So that's shown here. So this shows just very simply how insulin is going to bind to its receptor. And remember, we talked about this earlier in the course where we have these transporters. So it's insulin's job to get those transporters to the cell membrane. And then that lets the glucose get inside. And then the glucose can go through glycolysis to give you ATP if you need it. But if you don't, if you have anything extra, then the extra is used to make glycogen. So, and if you've made as much glycogen as you can, the rest of it gets converted to fat. So in question 10, what I wanted you to show is how can we use actually not just glucose, but alcohol and proteins to make fat. So this already, I guess, is part of that other diagram. So glucose goes to pyruvate. Pyruvate is converted to acetyl-CoA, and acetyl-CoA is the starting material to make fat. So this is kind of like a long, complicated process. In fatty acid synthesis, you make 3 fatty acids, 1 glycerol, and add the fatty acids to the glycerol, and you get triglycerides. Alcohol can do that too. Alcohol is broken down to acetyl-CoA, and again, you can use fatty acid synthesis. So if you have acetyl-CoA, you can use that to make fat. There are some proteins that we call ketogenic, and they form acetyl-CoA, again, which can form fat. There are glucogenic amino acids too. We'll talk about them later. That they're actually used to make glucose, not fat. So again, excess intake of carbohydrate, alcohol, proteins, and fat. So fat's not in here because it's already fat. That leads to synthesis of fat, and it's stored in our adipose tissue that we can call upon later. That's essentially the fed state. So the fed state, we ate food, we broke it down, and we used some of it for energy, and then the rest of it we stored as either glycogen in our muscle and liver or fat in our adipose tissue. So now, what happens if you go a long time without eating? So what if you wait 12 to 18 hours? Maybe you have dinner and then you go to sleep, and then you get up the next morning and you skip breakfast because you're late to class. So now what's happening is you're going to be either in post-absorptive or early fasting. So that's going to be up to, say, 12 to 18 hours after you eat. So that's our focus now. So on this graph, we are here. So again, the blood glucose concentration has to remain the same. So that means that we can have glycogen can break down and gluconeogenesis can happen. So the glycogen, that's the blue curve. And then the black curve right here is gluconeogenesis. So both of those are going to be happening to provide us with glucose that we're not eating. So so here's this student. He had dinner, he went to bed, and he was late to class and didn't have breakfast. So he's in short-term fasting state. So the resource he'll use is, remember, when he ate, was able to store glycogen, that's going to be broken down to glucose, and that's the fuel source for muscle cells. So the glucose doesn't leave, it stays in the muscle, and the muscle does not share it. That's different from the liver. The liver breaks down glycogen to glucose, and then the glucose can leave, and that's a fuel source for red blood cells in the brain and pretty much everybody else. And fat is broken down to a glycerol backbone and 1, 2, 3 fatty acids. That can be used by all cells except for red blood cells in the brain, which is pretty important. So that means you can't just have fat. You need to have some glycogen too, because the red blood cells in the brain need that. So again, so muscle glycogen breakdown in the muscle provides glucose that doesn't leave the muscle, powers the muscle. The glycogen breakdown in the liver provides glucose that leaves the liver for brain and other tissues. And adipose tissues provides fatty acids and glycerol that can be used by cells, except for red blood cells and the brain. So here, this is question 12. It kind of looks like the other table that I gave you, except now these complex molecules aren't food. They originally came from our food, but they're not directly from food. This is what we used our food to make. So this is storage glycogen, storage fat, and protein from our muscles. And these are all catabolic reactions, and they're broken down into smaller compounds, and energy is released. If I focus on fat here, when fat's broken down, so lipase enzymes will hydrolyze those 3 fatty acids, so you just have glycerol. Now, glycerol is a little bit special. It can get converted to pyruvate in the liver, and remember, the liver can undergo gluconeogenesis. Like I said, the liver and the kidney are the only 2 tissues who can do that. So we can get a little bit of glucose there. But nothing from the fatty acids, because what they do is they undergo a process called beta oxidation, where they're broken down into a whole pile of acetyl-CoA. And as we saw earlier, acetyl-CoA can go through the citric acid cycle and give us some energy. So that is what happens. So again, the triacylglycerols are hydrolyzed to fatty acids, and this is a process that if you're fasting, the glucagon will stimulate this. If you're exercising, if you're doing long-term exercise, epinephrine, you might know that as adrenaline, will stimulate that in the muscle. And then again, the glycerol goes to the liver because the liver can do gluconeogenesis, and you could get a little bit of glucose there. But most of the energy here, the fatty acids are broken down to acetyl-CoA, and they go through the citric acid cycle. And again, this occurs in the liver if you're fasting, or in the muscle if you're exercising. So let's talk about this pathway that we call beta oxidation. So the way that this works is, let's start up here. So in the fat chapter, we talked about what palmitate looks like. It has 16 carbons. So in this pathway, what happens is you go through a cycle, and every cycle you chop off 2 carbons as acetyl-CoA. So if you go through 1, 2, 3, 4, 5, 6, 7, because the last one just releases the acetyl-CoA. So what happens is you go through 7 cycles, and every time you do a cycle, you get 1 FADH2 and 1 NADH. So that means you get 7 of those, but you get 8 acetyl-CoAs. So the next thing that I ask you to do is, and here's a summary right here, is what's the ATP yield here? So if you go from palmitate to 8 acetyl-CoAs, you go through 7 of those cycles, where you go from 16, 14, 12, 10, 8, 6, and then the 4 is broken down into 2. So you get 8 acetyl-CoAs. Those have 2 carbons. So you go through 7 cycles, so 7 NADHs, 7 FADH2s, 8 acetyl-CoAs, and that means that you go through the citric acid cycle 8 times. If you get 2 CO2s, if you go around once, you get 16. That's all 16 carbons right here, all the way to CO2. And you get, again, if you get 1 ATP for 1 turn, you're going to get 8 ATP for 8 turns. You get 2 FADH2s for 1 turn, so you're going to get, you get 1 FADH2 for 1 turn, so you're going to get 8 for 8 turns. You get 3 NADHs for 1 turn, so you get 24 for 8 turns. So So again, remember that now, so you got some ATP, you got 8 ATPs here, but you have 7 NADHs, 7 FADH2s, 24 NADHs, 8 FADH2s, and remember 1 NADH is going to give us 3 ATPs, and 1 FADH2 gets us 2. So what you get is 7 times 3, 7 times 2, add this 8, 24 times 3, 8 times 2, and if you add it up, you should get 131 ATPs. So that's kind of a lot. So, so why is fat so fattening? So we remember carbohydrate is 4 calories per gram, and you get 38 ATPs as we just calculated. Fat, you get 131. And you might be tempted to say, well, the palmitate's just bigger. Well, that's true. But if I say, well, how many ATPs do you get per carbon? If I have 38 ATPs for a 6-carbon molecule, that's 6.3 ATPs per carbon. Oh, sorry, that should say 131. There's some complicated biochemistry that I didn't share with you. So 131 divided by 16, that's 8.2. So you still get more. It's not just the size of the molecule. And the reason for that is that if you look at these molecules, the palmitate has a ton of hydrogens. The glucose doesn't have as much. So it has some oxygen in here. So what that means is that if you have a lot of hydrogens, then you get a lot of NADH and FADH2. So you have a lot more NADH and FADH2 that can go to the electron transport chain and give you lots more ATP. So that's why fat has 9 calories per gram and carbohydrate only has 4. So oxygen is required to break down fats. This figure doesn't convey that concept very well because they're showing how you're breaking down the triacylglycerol. Here, the glycerol goes to pyruvate, and here the fatty acids get to acetyl-CoAs, and we go through many turns of the citric acid cycle. The NADHs and the FADH2 goes to the electron transport chain, and what they should show with it, what they don't, is the way you get water is that the oxygen accepts hydrogens to form water. So they should have drawn the oxygen here, but they didn't. But that's why you need oxygen, because oxygen is required to accept those hydrogens and electrons, and then it forms water. So one of my biochemistry books has an expression they call fats burn in the flame of carbohydrates. So what's interesting about this is that glucose forms pyruvate, and pyruvate can also form acetyl-CoA. But remember, way back, so go back and look if you don't remember, the very first step of the citric acid cycle is that acetyl-CoA reacts with something called oxaloacetate, or OAA, and that starts the cycle. And if everything is normal, the glucose will give you the OAA, and the glucose will give you acetyl-CoA too. But so will the lipid. So you'll get the OAA and the acetyl-CoA you need to start the cycle. So as long as you have both carbohydrate metabolism and lipid metabolism, and as long as they're both normal, everything's fine. But sometimes carbohydrate metabolism isn't normal during untreated diabetes. And if you don't have insulin, glucose is there, but it can't go in the cells. Remember, insulin, its job is to have that carrier so the glucose can get inside the cell. Starvation, you don't have any glucose available in the diet. And there's some very sort of trendy keto diets, Atkins, low carb, high protein, high fat diets. And you might not be fasting or starving, but you're not eating any carbohydrates, so you have no glucose. So what happens then? Well, what happens then is if you don't have any glucose, what happens is amino acids will be converted to OAA, and then what happens is OAA forms pyruvate, and then this is gluconeogenesis, and that forms glucose. So this is the source. So if you're starving, these amino acids come from your muscle protein. But if you're on a high protein diet, this comes from the amino acids and the protein in your diet. And then all of this is being used to make glucose. So what that means is that you don't have any OAA. So the fat is being broken down to acetyl-CoA, but you have no OAA. So that means if you can't do the first step of the cycle, you can't finish the cycle. So acetyl-CoA kind of piles up. And, oh my God, I went the wrong way to say it. Sorry. Okay, so, and it forms something called ketones. And that's why this is called the keto diet. So it forms something called ketones. And this is how it works. So again, the fatty acids break down acetyl-CoA, and we want the acetyl-CoA to go to the citric acid cycle. But if you don't have OAA, because the OAA is being diverted to gluconeogenesis, then you have a pile of acetyl-CoA and forms ketone bodies. So one of the ketones bodies is called acetoacetate, one's called beta-hydroxybutyrate, and also there's acetone, which you might have heard of. Acetone actually is nail polish remover and smells like it. So what happens is you get ketones in your diet, in your blood, and then that shows up on your breath. So if you go to websites that talk about the keto diet, people would complain about having bad breath, and that's why, because the ketone bodies, one of them is acetone, and it smells bad. So again, if OAA is not available to react with acetyl-CoA, then you form these ketone bodies instead. Now, some tissues actually can use them for energy. So you have high concentration of ketone bodies. The brain really prefers glucose, but if it can use ketone bodies if it has to, and the muscle can use ketone bodies. But if you can't survive on that indefinitely, ketosis will eventually cause coma and death because ketone bodies are acidic and they cause loss of sodium ions and metabolic acidosis. So your blood gets too acidic, and that's not compatible with healthy life. Now let's talk about protein. So the body's priority is use of protein for building and repair of body tissues. And in terms of energy, it uses fat and carbohydrate, assuming that it has fat and carbohydrate. So protein is saved for metabolic functions that can't be performed by other compounds. But you can use it for fuel if you have to. If you're not eating a lot, or if you're not eating any carbohydrate, then the body is actually going to use protein, either protein from your diet or protein from your own body, because the brain really, really likes to have glucose. So in question 17, I asked you to talk about what happens here. So in an amino acid, we talked about how amino acids have nitrogen. So what you have to do is you have to strip off the nitrogen as NH3, and that goes through something called the urea cycle. And the carbon skeletons, that's kind of what's left. What happens to them depends on what you need. You can actually bring this through a catabolic pathway and make ATP, or you can take it through gluconeogenesis in the liver. You can make acetyl-CoA, and if you have too much acetyl-CoA, maybe you'll be making ketone bodies. So it just depends on what is available. So the removal of the amino group as ammonia happens first, and then the carbon skeletons go other places depending on what you need. So let's go back right here. So this is the urea cycle. So the urea cycle, what happens is you can't just excrete. NH3 is ammonia, and that's that same product that you clean with. So and that's toxic to the body. So we can't have ammonia. So what happens is there's something called the urea cycle, and what it does is it converts this to a compound called urea, which is not toxic. So that leaves the liver and goes to the kidney, and then the kidney excretes the urea in the urine. So anything there are other nitrogen containing compounds in the body. So if that nitrogen's not used, then you just excrete it. So you can look at someone's urine and see how much nitrogen they're excreting and see if they're in nitrogen balance or not. The catabolic processes that make glucose and you're making ketone bodies. So you'll make ketone bodies and the brain isn't happy about it, but it will use it if it has to. So those glucogenic amino acids I told you about. So some amino acids can get converted to pyruvate, and then this blue arrow right here shows gluconeogenesis. But the ketogenic amino acids form acetyl-CoA, and notice that there is no arrow in this direction. So once you form acetyl-CoA, you can't form glucose. And remember, fatty acids will form acetyl-CoA, and alcohol will too. So at this point, if you have acetyl-CoA, the only thing you can do is you can send it through the citric acid cycle if you have OAA. And if not, you make ketone bodies. So the amino acids with at least 3 carbons. So that's the magic number. So if you have 3 carbons, remember pyruvate has 3 carbons. If you have 3 carbons or more, but if you have at least 3, you can go to pyruvate and you can form glucose. So again, that blue arrow is gluconeogenesis. But if you only have 2, then you go through excuse me, the citric acid cycle if OAA is available, or you form ketone bodies if it's not. So these amino acid carbon skeletons have other entry points. So the carbon skeletons, we remember, have 20 amino acids, and they're all different. And some of them can form acetyl-CoA, some can form pyruvate, some of them actually can feed right into the citric acid cycle itself. So there are lots of different places your amino acids can go. And again, the balance is going to be what does the body need at that point. So the last thing we'll just talk about really briefly is the fasting state, which lasts from 18 hours to 2 days. And what's happening here, so now I'm in this box right here. So our glycogen is probably going to be gone at this point because, again, your liver and your muscle only store so much of it, so it's probably going to be gone. So you're going to really ramp up gluconeogenesis. And again, you can't get it from fat, so you have to get it from amino acids. So what happens in the short term is if you look here, so again, muscle protein will form amino acids. They can go to the liver, and the glycerol from the backbone, that can go through gluconeogenesis, but the fat can't. So notice that the fat's not over here. The fat forms ketone bodies. And again, the ketone bodies are an alternative fuel source. So some tissues can use the fatty acids as they are, but if you send them to the liver and they go through beta oxidation and then you can't go through the citric acid cycle, that's when you get the ketone bodies. So gluconeogenesis occurs in the liver and the kidney if glycogen is gone. Glucagon is going to stimulate that. So the sources are glycerol, lactate, and amino acids. So at this point, you're starting to break down body protein, and that's really not sustainable. So what happens next then is if you don't eat after that, then the body switches over because it's not sustainable. And so the body switches over and stops breaking down protein and just exclusively does fat. So how long you can survive, it depends on how much fat you have. So some of these studies have been done in hunger strikers. So in the times euphemistically referred to as the Troubles in Ireland, there were members of the IRA that, as a protest, went on hunger strikes for 40 days, and a lot of them died. So you can't go this long without food. So that was our whirlwind tour through Chapter 7, and I hope that this is helpful. And if it's not, come to my office hours, and I'd be happy to answer any questions that you have. Thank you for your time and attention, and have a good day.